Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing ICMR BRET question paper for the subject biochemistry. This is section B of the question paper which was conducted in the year section C of the question paper which was conducted in the year 2024. The cross disciplinary I have already discussed. And if you want still previous year question papers of ICMR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, select for ICMR and you will find them there. And if you are new to my channel, please do like, share and subscribe. Now, before coming to the first question, we will see about the ATP dependent pathway for protein degradation by ubiquitin. So, it is by three steps, that is ubiquitin activation, then conjugation and ligation, and you have E1, E2 and E3 enzymes. So ubiquitin activation by E1, that is ubiquitin activating enzyme. So this enzyme, it will use ATP to form a thioester bond between the C-terminal carboxyl group of ubiquitin and the cysteine residue of E1. See the bond formed. Then there is the ubiquitin conjugation, that is by the enzyme E2, that is ubiquitin conjugating enzyme, and this ubiquitin from E1 enzyme is transferred to E2 enzyme via a trans-esterification reaction which result in the formation of a thioester bond between the with the cysteine on the E2 enzyme. Okay, so E1 is also having a cysteine, E2 is also having a cysteine. They form the thioester bond with the cysteine on the E2 enzyme. Now there is the another enzyme, that is E3, which is involved in ubiquitin ligation, or that is E3 is known as ubiquitin ligase. Now the E3, it will recognize the target protein. This is the target protein having a lysine residue as well as what is called as degron. Degron is a short amino acid sequence within the target protein that act as a tag for its degradation by the ubiquitin proteasome system, and this degron, it is recognized by E3 ligase. Okay, now this E3 ligase, it will transfer the ubiquitin from the E2 enzyme to this target protein. So what will happen is, it will create a isopeptide bond between the glycine residue at the C terminus of ubiquitin and the lysine residue in the target protein. And upon this repetition of this step, a chain of ubiquitin molecules will be added to the target proteins, which will be recognized and will be directed to the 26S proteasome for degradation into smaller peptides and amino acids which will be recycled by the cell, and this is an ATP dependent process. So this is what is happening in the ubiquitin proteasome ATP dependent degradation of protein pathway. Now come to the question. The ATP dependent pathway of protein degradation in eukaryotic cells is quite different, involving the protein ubiquitin. Ubiquitin is covalently linked to protein determined for destruction. Yes, we already saw that, via an ATP dependent pathway involving three separate type of enzymes. Three enzymes were there, E1, E2, E3. Identify the correct set of three separate types of enzyme. So what are these enzymes is your question. So E1 activating enzyme, E2 conjugating enzyme, and E3 ligases. So answer is option 3. Then next one, in chronic kidney disease, hyperkalemia is often a concern due to impaired renal excretion. Yes, chronic kidney disease, there is impaired renal excretion which will result in hyperkalemia. Which hormones decreased activity exacerbates this condition? Now hyperkalemia in chronic kidney disease, if the person is having deficiency of some hormone, what hormone will cause or will amplify this hyperkalemic condition? So if you know the aldosterone hormone, it is a major hormone that regulates the K plus balance by promoting its excretion in the kidneys. So when kidney function is reduced in chronic kidney disease, the kidney is less able to excrete K plus from its K plus through urine, okay, and maintain the balance of K plus in the body. So a hormone like aldosterone, if it is produced in decreased amount or in decreased concentration, it can also cause hyperkalemia, okay, or it can exacerbate in chronic kidney disease. Then which one of the following is an experimental method for determining three-dimensional structure of a protein? So it is X-ray crystallography. This threading, ab initio, homology modeling, they are not experimental methods, they are computer-based bioinformatic methods for determining three-dimensional structure of proteins. So all the three I have discussed in cross-disciplinary, which was conducted in the same year. Then choose a compound with the best docking score. Now docking, it is a molecular modeling technique that is used to predict how a protein interacts with a ligand. And this is mainly used in drug discovery technique, that is to predict how the potential drug will bind to the disease causing protein. Okay, that is what is you do in docking studies. Now what is the best score they have asked for? So the best score is the one with the most negative binding energy or more negative value, because that will indicate the more favorable and stable interaction between the ligand and its protein. So here the more negative value is -9, so that is the best docking score. Then which algorithm is used for global alignment? It is Needleman-Wunsch, Smith, Waterman and Alschool. All these are local alignment, and day off, it is a substitution matrix used when you are doing protein sequence alignment, and it will tell you the probability that one amino acid is replaced by another. That is what day off is telling. Then next one, in FASTA format, the definition line start with this symbol, that is option 3, greater than symbol. Then the PDB ID or PDB code is of dash character and is represented as dash. So PDB is a 4-character alphanumeric and it is serve as a unique accession number for every molecular model that is deposited in the PDB. Now this 4 character, the first character will be a digit from 1 to 9. The remaining 3 characters can be either a digit 0 to 9, or it can be any uppercase letters, that is A to Z in uppercase. So that is the PDB code. So it is 4 and alphanumeric. Then when aligning sequences that are highly divergent, best results will be obtained with which substitution matrix? Now highly divergent means these sequences, they have undergone significant changes over time, and they will be showing low similarity compared to other sequences. Then there will be no conserved nucleotides or amino acids present in them, and these sequences would have accumulated many mutations over time, and there may be difficulty to identify homologs of this sequence and infer functional relationship. So these are highly divergent sequences. So the best substitution matrix for highly divergent sequence is PAM 250. Now PAM 250 is suitable for sequences with low similarity, because it is a higher PAM value. PAM 250 means high PAM unit value, which means more substitutions per site. So it is suitable for highly divergent sequences. Now if they have asked a question for BLOSSOM matrix, then you will have to go for BLOSSOM 30 or BLOSSOM 45. The next one, transport proteins associated with facilitated diffusion process include. So facilitated diffusion, it is a passive process. And in this method, the ions or the molecules move down their concentration gradient with the help of specific transport proteins, because these molecules, they are large enough that they cannot cross the cell membrane on their own. So uniporter, it is a facilitated diffusion one, and it is a type of transport proteins that will facilitate the movement of one type of molecule across the cell membrane down its concentration gradient without use of ATP. Now pump motor, it is for active transport. Ion channels, again facilitated diffusion process. They create pores or channels through the lipid bilayer of the cell membrane, and the ions, they will move through these channel passively. Then there are transport motors, again with active transport. So A and C are the right answers. So option 3 is the answer. Then plants that take up high amount of metal elements from soil and store them in their aerial tissues are known as hyperaccumulators as well as metallophytes, not chelators and reducers. So metallophytes, they are plants that survive in soil rich in heavy metals, and hyperaccumulators, they are type of metal or group of metallophytes that accumulate high concentration of metal in their tissues and without showing any signs of toxicity. So A and B both are right, making option 3 as the right answer. Then C2 oxidative photosynthetic carbon cycle. Okay, this I discussed when during cross-disciplinary conducted in the same year. The same question somewhat related to this was asked there also. So C2 oxidative photosynthetic carbon cycle is nothing but your photorespiration and photosynthetic carbon oxidation cycle, not Calvin benzene and the reduction of pentose phosphate pathway. So A and C are correct. Answer is option 1. Then leaves maximize light absorption by continually adjusting the orientation of the leaf lamina is called solar tracking. Now what is light channeling? Light channeling, it is the ability of some plants to direct light to specific areas, such as in case of trichomes of the leaves. They will channel the light to specific regions. So that is light channeling. Then what is sunflex that you see in this figure picture over here? Sunflex means they are moving patches of sunlight that penetrate the gaps in forest or plant canopy. So because of this long, huge, tall trees, light will penetrate only through small gaps between these trees and it will reach the ground. So in the ground there will be plants which will not be getting direct sunlight always, just because they are not tall enough. So because of this sunflex, the ones which are in the shaded portion of these huge trees, they will get light, and thereby they can increase their photosynthetic activity. So that is sunflex. And fluorescence, it is nothing but emission of light by plants after absorbing light energy. Then next one, secondary macronutrients include. So secondary macronutrients are calcium, magnesium and sulfur, and they are called secondary macronutrients because the plant often get them in sufficient amounts from the soil. And they are required in lesser amounts than the primary macronutrients. The primary macronutrients are your nitrogen, phosphorus and potassium. So the answer is calcium and magnesium only. The next one, two peptides are subjected to column chromatography at the pH of mobile phase. Peptide A has a net charge of -3 due to more glutamic acid aspartic acid residues. Peptide B has a net charge of +1. Peptide A will elute first in cation exchange column. Okay, cation exchange column means it will use a negatively charged resin. Now peptide A is negatively charged. It will not bind to the cation exchange resin. It will elute first. So peptide A will elute first. Statement 1 is correct. Now peptide B will elute second in anion exchange column. Anion exchange column, they have positively charged resin. So peptide B, it will not bind to the anion exchange resin and it will elute first, not second. So statement 2 is incorrect. So statement 1 is correct, statement 2 is incorrect. That makes option 3 as your answer. Then next one, PCR can be used for in vitro amplification of genes. That is correct. PCR can be used to introduce site-directed point mutation in a cloned gene. That is also correct. So that makes option both statement 1 and 2 are true. Now how this PCR introduce site-directed point mutation in a cloned gene, I will tell you. Suppose they ask for the this 2025 question paper. So I'm just explaining how PCR does this process. So here you have a gene in the plasmid. This dark portion one, that is the gene in the plasmid. That is the target site for mutation. Okay. So this is a gene that is cloned in a plasmid. So site-directed mutagenesis, you know, it is a method for altering the nucleotide sequence of a gene at a specific location, and it can be done through point mutations, that is substitution, deletions, etc. Now what you do first here is you synthesize a complementary oligonucleotide primers. and the primers should be such that they contain a point mutation at the desired region. So these are the oligonucleotide primers complementary to this particular target gene, and they are having a point mutation. Now you perform PCR amplification using these primers and this DNA as this, or the cloned plasmid DNA as the template. And you should use high fidelity DNA polymerases, because only if you use the normal standard polymerase for PCR, then there are chances that these standard polymerases, they can introduce random mutation throughout the amplified DNA. But if you use high fidelity DNA polymerases, only the specified mutation will be present in the final amplified DNA product. Now after completion of the PCR, you have to remove this template plasmid DNA, or the original plasmid DNA you have to remove. You need only the one which is having your point mutation. Now this plasmid DNA, the template plasmid DNA, how did you get? You got it isolated from an E. coli. So when the E. coli replicates, it will replicate this plasmid DNA, and this E. coli, it is having a tendency to methylate its DNA strands upon replication at the GATC site by the help of the enzyme DAM methylase. So here you are doing PCR, you don't add any methylase there to methylate specific DNA sequences. So whatever parent or template plasmid DNA is present, they will be methylated, whereas the amplified product will not be methylated. So this template plasmid can be treated with DPN1. DPN1, it will digest the methylated plasmid DNA. So what do you get the result? The methylated plasmid DNA will be digested of the remaining one which is having the point mutation, that plasmid will be present. Now this plasmid you take and transform into competent E. coli cells. The cells will replicate the plasmid. Now you plate these E. coli onto an antibiotic media for selection. So the ones which have taken up the plasmid, they will grow on this antibiotic media, whereas the others which have not taken up the plasmid will die off. Then you isolate plasmid from individual colonies and send them for sequencing, so that after sequencing you can confirm that the desired point mutation have been successfully introduced. So site directed mutagenesis with PCR. Okay. Now come to the next question. Okay, match the list. So here you can see two terms, spectroscopy and spectrometry. So spectroscopy, it is just the scientific study of the interaction between matter and the electromagnetic radiation. It just tells you how the sample absorb or emit or scatters light of specific wavelength. It doesn't give any result. Quantitative result is not obtained by spectroscopy. It is simply just a theoretical approach, and it is a non-destructive method. Your sample is not destroyed when you conduct spectroscopy. Example is IR spectroscopy. It is uses infrared light to identify functional groups in a molecule. Okay, this functional group is there, that functional group is there. All these you can identify by infrared spectroscopy. So A2 wavelength will be the match for spectroscopy wavelength. Then what about spectrometry? Spectrometry, it is the practical application of spectroscopy, and it uses instrument like spectrometers, and it will measure the intensity of light interacting with the sample, and it generates result that can be used for quantification. That is, it gives measurable data. Example is the case of mass spectrometry. It uses it generates multiple ions from the sample. That is, it will destroy your sample. It will generate multiple ions from the sample, and then it separates them according to their specific mass to charge ratio, and then record the abundance of the ion in the sample, and provide information regarding the molecular composition of the sample. So that is what spectrometry is doing. So C will be 4, that is ionization. So A2C4 will make option 4 as your answer. Okay, what about the rest? Chromatography, it will be pH. Based on difference in the pH, you can elute through the chromatographic column. And crystallography, it is X-ray crystallography that is used for protein structure studies. So 4 is the answer. Then next one, which of the following restriction endonucleases are 4-base cluster and leads to blunt end? And this 4-base cluster means they recognize 4 nucleotides. Blunt ends means they will not be like this. If you cut at a particular restriction enzyme, you will get this type of cut, that is blunt end. And if you get these overhangs like this, it is cohesive ends. So you have to see, for blunt end means they will cut directly in the center of the recognition sequence usually. Okay, we will see one by one. So ALU and A3, it is 4-base. See here, 4 bases are there, AGCT and GGCC. They cut over here in the center, and you will get blunt ends like this. So first one is the right answer. Then coming to the next, it is AVA1 and HHA1. You can see AVA1 is more than 4 bases, whereas HHA1 is 4 bases, but HHA1 is not cutting directly in the center. It is also generating sticky, and I think it will cut here, here, and AVA1 also cuts here. So it will generate sticky ends. Then you have got the other one, that is BAM-H1 and HIN3. They are also not 4 nucleotides. BAM-H1 will cut here, HIN3 will cut here. So again it will generate sticky ends. KPN1 and HPA1, again KPN1 I think it is cutting here, and HPA1 it is cutting here, again sticky end generation. Oh sorry, HPA1 is a blunt end cutter. Okay, it will cut here, not here. It will cut in the center. So HPA1 will generate blunt ends, but KPN1 will generate sticky ends. So that is also not the answer. Then next one, match list 1 with list 2. Okay, confocal microscope, that is for actin fibers. A3. Okay, A3 we have got in option 1 and okay 3. Okay, 3. Okay, by mistake I marked 3 also wrong. Okay, we will see. A3 is A3. Then B, transmission electron microscope for studying virus structures. Okay, B1 in 1 and 3 both are correct. Then phase contrast microscope is for unicellular organism. Okay, so 3 is wrong, 1 is correct. And atomic force microscope, it is for prion fibrils. So D2. So A3, B1, C4 and D2 is the answer. Then next one, okay, protein primary structure. Protein primary structure you can determine by tandem mass spectrometry. A3, oh, that makes option 3 itself as the right answer. Secondary structure by circular dichorism, yes. Tertiary structure by nuclear magnetic NMR, yes. Protein quaternary structure by gel filtration, that is also correct, because in gel filtration the native structure of the protein is maintained as such. When you pass through the gel filtration chromatography, the native conformation of the protein is not changed. So D2. So A3, B1, C4 and D2 are the answers. So that's it now. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel, and if you want still previous year question papers of biochemistry being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist select for ICMR and you will find them there. Thank you.